Yeah, so we'll begin this talk with a talk by Dr. SIDDHARTH NARENDRAN on a very exciting talk. He's doing some great work at Arvind Coimbatore, and it's been a pleasure to see him grow and work in this area. So over to you, SIDDHARTH.
Thank you. So my talk is titled Mechanotransduction, a paradigm shift in the pathogenesis of AMD. So this is a talk I presented last year. Over the past 1 year, I've added more exciting data to this presentation. So the two hallmarks of AMD are, one is aging, which is the hallmark risk factor, and two is the sub-RP deposits or drusen's, which is just the pathological hallmark. So identifying where these two hallmarks converge, a pathway where these two hallmarks converge could be a potential target to treat age-related macular degeneration. So the process is called mechanotransduction, was introduced 2 decades back, and they too play an important role in both aging and in disruptions of the extracellular matrix. So what is mechanotransduction? So mechanotransduction is the phenomenon by which mechanical signals are converted into biochemical responses. Dysregulation of this process has been implicated in several diseases. For example, dysregulations to flow in atherosclerosis, to stretch in cardiac hypertrophy, and to confluence in cancer. An RP being a confluent monolayer, what is the mechanical stimuli to which it is exposed? So cells also respond to changes in the mechanical stimulus of the substratum on which they sit. So cellular homeostasis is influenced by changes in the substrate stiffness and in the components. As we know, the substratum on which the RP sits is one of the pathological niches in AMD. So the RP, which usually sits on its basement membrane, comes to sit on these sub-RP deposits in macular degeneration. So our hypothesis was alterations in the mechanical properties of the Brooks membrane induced by these sub-RP deposits perpetuate inflammation and subsequent RP degeneration. How did we go about testing these hypotheses? First was to study what are the mechanical changes which are happening at the Brooks membrane in AMD. The second was to study what are the effects of these changes on the RPE. And the third was to study what is the signaling pathways and if we could identify any putative therapeutic targets. So the first part was to study the biomechanics of the Brooks membrane. This we did by using an atomic force microscopy. So we had 3 groups: early AMD, normal eyes, and geographic atrophy eyes. So these are all donor eyes with these diagnoses. So this is again interesting. So we measured the Young's modulus using atomic force microscopy. So except the macular area, all other areas, the Young's modulus was insignificant. So the submacular region in geographic atrophy was significantly stiffer compared to early AMD or normal macula. So what is the functional relevance of these biomechanical changes? So this is a quantitative value to understand what is the qualitative influence of this. So we plated RPE cells, human RPE cells, isolated from a single healthy donor onto the Brooks membrane of 2 different eyes. One was a normal eye and the other was an eye with geographic atrophy. So and again we looked at the RP adhesion using scanning electron microscopy. So the white hyperreflective dots you see are RPE cells. As you can see here on the left bottom, RPE adhesion was significantly diminished only in the macular region in geographic atrophy, but not in the peripheral region. So excited by these findings, we then wanted to look at mechanotransduction. An analog I used to understand mechanotransduction is the microphone. So in the microphone, mechanical signals are converted into electrical signals, while in mechanotransduction in the cell, these mechanical signals are rather converted into biochemical signals. So here we are interested in studying the microphone. So the microphone is the instrument which is converting these mechanical into electrical signals. Similarly, mechanotransduction is the signaling pathway which converts the mechanical signals to biochemical signals in the cell. So again, mechanotransduction involves 2 main components. One is the mechanosensor and then the mechanotransducer. So this is again, we can compare it to the microphone. So the mechanosensor senses the changes in the mechanical stimuli and then converges into the mechanotransducers which act on these changes. So we needed a starting point. So unfortunately, mechanosensors are multiple transmembrane domains, and so they are, we do not know which one to target first. However, irrespective of the mechanosensors, they converge on 2 main mechanotransducers called YAP and TAS. This is something like this. So we thought we will start with YAP and TAS and see if they play a role in macular degeneration and RP cellular homeostasis. So again, any complex new hypothesis would require a triangulation of cell culture, human data, and animal model. So we started to study the effect of deleting these YAP and TAS expression in vivo and in vitro systems. First, in cell culture, the knockdown of these YAP and TAS proteins reduced RPE viability and increased RPE inflammation. And I think this is the most interesting data we have. We studied by conditionally knocking down YAP and TAS expression only in the RPE in mice. And these mice developed degeneration of the RPE as seen in these fundus photographs and in these optical coherence tomography images and in this RPE flat mounts. So this is the morphology of the RPE in these YAP and TAS knockout mice. And this is the morphology in AMD human donor eyes. What was interesting is these mice also developed features of choroidal neovascularization. So this is the only model, animal model, which produces both phenotypes seen in AMD. And now we believe that differential expression of these two proteins could be driving phenotypic diversity in AMD. So the next was to study the human data. So we looked at the expression of these two proteins in GA. As you can see here, RNA protein levels were significantly reduced in the RPE in donor eyes with geographic atrophy. So what makes it even more intriguing is that there is this rare disease called Swainson's chorioretinal atrophy. And Swainson's chorioretinal atrophy, the mutation is in this mechanotransduction pathway, and RPE degeneration is the only pathological hallmark in this. We were lucky to get a donor eyes with Swainson's chorioretinal atrophy, and we looked at the mechanotransduction pathway, and it was affected similar in geographic atrophy. So this is our conclusion, that the sub-RP deposits disrupt normal mechanotransduction pathways, and this leads to decrease in the mechanotransducers YAP and TAS, resulting in RP inflammation and degeneration. For the past 3 decades, identifying the components of the drusen and targeting them has been the predominant therapeutic strategy for AMD. We believe that this new hypothesis would create a paradigm shift in our understanding of macular degeneration, a shift from the existing biochemical hypothesis to a mechanochemical hypothesis. Thank you.
Yeah, so great talk, Siddharth. So going forwards, what therapeutic implications do you see from pharmacological aspect?
So this
So
is
since you're
interesting, sir.
completely changed the ball game now.
Yeah, so it's interesting because we learn from metastasis. So we learn from metastasis. So in metastasis also they want the cells to remain adhere and not go away to to seed others. So metastasis, they try to target cellular adhesion. So similarly, what we are also trying to improve cellular adhesion. RP. This is also, I think, a reason why many of the RP transplantations fail. That even though the cells are good, the scaffold is not great. So we have to also target the scaffold on which the RP lies.
Any questions? Thank you, Siddharth. Exciting talk.